Welcome back. Today, we're digging into a stack of material all about the tools of genetic engineering. We want to pull out the key knowledge, the stuff that lets scientists go from just mapping genes to actually editing them.
Absolutely. It's a fascinating journey.
And it really seems to kick off properly around 1971. That's when Dana and Nathan showed how certain bacterial enzymes could precisely cut DNA.
That was definitely the beginning of what we call the recombinant DNA era, and things moved incredibly fast.
Yeah, seems like it. By 1978, Nathan, Smith, and Arbor had the Nobel Prize for basically giving us the tools for gene splicing.
It really changed the game. Before that, studying a single gene within a whole genome was, well, incredibly difficult.
Like finding a needle in a haystack.
A huge haystack, yes.
Yeah.
This technology, for the first time, let researchers reliably identify, cut out, and study just one specific DNA sequence. It was like inventing the index for the book of life.
Okay, so that's our goal here, to map out this molecular toolkit for you. We're talking about recombinant DNA technology, basically joining DNA from different places artificially.
Right, and we could break it down maybe into four main areas.
Sounds good. What are they?
Well, first, the basic tools themselves. Then, the strategies people develop for cloning DNA. After that, how you actually analyze what you've made. And finally, the ways we manipulate DNA inside living organisms.
All right, let's start with those foundational tools. If you want to join DNA, you first need to cut it, right?
Exactly. And that's the job of restriction enzymes, sometimes called molecular scissors.
Molecular scissors, okay. And these came from bacteria.
Yeah. Interestingly, they're part of a bacterium's defense system against viruses, bacteriophages specifically.
Ah, so the bacteria use them to chop up invading viral DNA?
Precisely. They restrict the virus's ability to replicate. And the key thing for us is their precision.
How so?
Each enzyme recognizes a very specific short sequence of nucleotides, the restriction site, and cuts the DNA's backbone right there, always at the same spot.
And that predictability is crucial.
Absolutely essential. It means you get consistent, identical DNA fragments every time you use that enzyme.
Okay, I've heard about sticky ends in this context. What's that about?
Right, so it depends on how the enzyme cuts. Many of these restriction sites are palindromic.
Like Madam, I'm Adam. Reads the same forwards and backwards.
Sort of, but on the DNA strands. It reads the same 5' to 3' on both strands. Now, an enzyme like E. coli cuts this sequence unevenly, making staggered cuts.
Okay.
And that leaves these little single-stranded overhangs on each end. Those are the cohesive ends, or sticky ends.
Sticky because they can stick to other ends.
Exactly. They're complementary. So an end from one piece of DNA can base pair, or anneal, with a complementary sticky end from another piece of DNA.
As long as they were cut by the same enzyme.
That's the key. If they were cut by the same enzyme, the ends match perfectly, then you just need the molecular glue. DNA legs. This enzyme comes in and seals the gaps in the sugar phosphate backbone, covalently linking the fragments together. And boom, you have your recombinant DNA molecule.
Okay, cut with restriction enzymes, anneal the sticky ends, glue with ligase. Got it. Now you have this new DNA molecule, but you need to copy it, right? And maybe get it into a cell.
Right. That's where cloning vectors come in. Think of them as delivery vehicles for the DNA.
Delivery vehicles? Like what?
They're DNA molecules themselves, ones that can accept foreign DNA fragments. Crucially, they can also replicate independently inside a host cell, like a bacterium.
What makes a good vector? What properties does it need?
Well, several key things. It needs restriction sites, often a whole cluster of them called a multiple cloning site where you can insert your DNA. It needs an origin of replication, or ORI, so the host cell's machinery will copy it.
Okay, makes sense.
It also needs a selectable marker, something like an antibiotic resistance gene.
Why is that important?
Because getting the vector into the host cell, the process called transformation, isn't very efficient. The marker lets you kill off all the cells that didn't take up the vector. Only the ones with the vector survive on the antibiotic plate.
Clever. Anything else?
Often, they'll also have sequences nearby that allow for easy DNA sequencing later on.
And the most common type of vector, especially early on, was the plasmid.
Yes, bacterial plasmids, those small, circular, extrachromosomal bits of DNA. They're relatively easy to work with. We usually get them into bacteria using either a heat shock treatment with calcium ions or a zap of electricity called electroporation.
But you mentioned selection. How do you know if the plasmid that got in actually contains the piece of foreign DNA you wanted to insert, not just the original empty vector?
That's where another clever trick comes in. Blue-white screening. It uses a gene called lacZ.
The one involved in lactose metabolism.
That's the one. It codes for an enzyme, galactose. In many vectors, the multiple cloning site is deliberately placed right in the middle of this lacZ gene.
Okay, so inserting your DNA breaks the gene.
Exactly. If you successfully insert your foreign DNA, you disrupt the lacZ gene. It can't make a functional enzyme anymore.
And how does that lead to blue or white colonies?
You grow the transformed bacteria on a plate containing a special chemical substrate called xGal. If the plasmid doesn't have an insert, lacZ works, the enzyme is made, it cleaves xGal, and the bacterial colony turns blue.
But if your insert is there,
then lacZ is broken, no functional enzyme is made, xGal isn't cleaved, and the colony stays white.
so the white colonies are the ones you want, the ones with your recombinant plasmid.
Precisely. It's a really neat visual way to identify successful cloning events. White is right in this case.
Okay, that's plasmids. But you mentioned they have limits.
Yeah, size limits. Plasmids are generally only good for relatively small inserts, up to maybe 25,000 base pairs or 25 kilobases.
Which isn't enough if you're trying to study large genes or, whole genomes.
Right. As ambitions grew, especially towards mapping entire genomes like the Human Genome Project, we needed vectors that could carry much larger chunks of DNA.
So what came next?
Things like phage vectors, based on bacteriophages, could hold a bit more, maybe 45 kilobands. But the real workhorses for large fragments became BACs and YACs.
BACs and YACs.
Bacterial Artificial Chromosomes, or BACs. They can handle inserts of 100 to 300 kilobases. And even bigger were Yeast Artificial Chromosomes, YACs, which could carry up to 1 million base pairs, a megabase, or even more.
Wow. Okay. 1 million base pairs. That's substantial.
Absolutely essential for tackling large genomes. And these larger vectors were key for building DNA libraries.
They did libraries, like a library of genes?
Kind of. They're collections of cloned DNA fragments from an organism. There are two main types, and the difference is really important.
Okay, what are they?
First, you have genomic libraries. These aim to contain fragments representing the entire genome of an organism, all the coding sequences, introns, regulatory regions, everything. It's like a complete, unedited blueprint.
The whole shebang.
The whole shebang. Then you have cDNA libraries. These are quite different. cDNA libraries are made starting from messenger RNA, or mRNA, isolated from a specific cell type or tissue at a specific time.
So only the genes that are actively being transcribed into RNA.
Exactly. You use an enzyme called reverse transcriptase to make DNA copies. That's the C in cDNA for complementary DNA of the mRNA molecules. So a cDNA library gives you a snapshot of gene expression, which genes were switched on in those particular cells at that moment.
That seems incredibly useful. If you want to know what makes a liver cell different from a brain cell or what changes in cancer.
Precisely. You'd compare their cDNA libraries. Now, finding your specific gene of interest within these massive libraries used to involve screening them with a labeled probe, a complementary piece of DNA or RNA that would stick to your target sequence. It was laborious.
Sounds like it. But then came another revolution, right? PCR.
Oh, absolutely. PCR? Yeah. The Polymerase Chain reaction, developed by Kary Mullis, for which he got a Nobel. PCR changed everything because it allowed amplification of specific DNA sequences without needing to clone them in cells first.
And it works on tiny amounts of starting material.
Vanishingly small amounts. DNA from a single cell, a fossil fragment, a drop of blood, and it's fast.
How does it work? What do you need?
You need the DNA template you want to copy, of course. You need the building blocks, deoxynucleotide triphosphates, or DNTPs. You need short DNA primers that flank the target sequence you want to amplify, magnesium ions, and crucially, a special DNA polymerase.
Special how?
It has to be thermostable, able to withstand high temperatures. The most famous is Taq polymerase.
Taq. From where?
From Thermus aquaticus, a bacterium found living in hot springs in Yellowstone. Because it lives at high temperatures, its enzymes, including its DNA polymerase, don't get destroyed by the heat needed in the PCR cycle.
Okay, so walk us through that cycle. It repeats over and over in a machine, right?
Yep, in a thermocycler. It's basically 3 steps. First, denaturation. You heat the mixture up to about 92, 95 degrees Celsius. This melts the double-stranded DNA template into single strands.
Separates the strands?
Right. Second, annealing. You cool it down, maybe to 50, 65 degrees Celsius. This allows the short primers to bind, or anneal, to their complementary sequences on the single-stranded template DNA.
So the primers mark the start and points for copying.
Exactly. And third, extension. You raise the temperature again, usually to around 72 degrees Celsius, which is the optimal temperature for Taq polymerase. Taq then synthesizes new DNA strands, starting from the primers and extending outwards, using the template strand as a guide.
And then you just repeat those 3 steps.
Over and over. Denature, anneal, extend. Each cycle takes only a few minutes, and it doubles the amount of the target DNA sequence.
So it grows exponentially.
Exponentially. After 20 or 30 cycles, which takes just a couple of hours, you can get millions or even billions of copies from just a few starting molecules. It's incredibly powerful.
And there are variations on PCR, too.
Oh, yes. For instance, reverse transcription PCR, or RT-PCR, that starts with RNA, uses reverse transcriptase to make cDNA first, and then does PCR. It's great for measuring gene expression levels.
Comparing how much RNA for a certain gene is present.
Right. And then there's Quantitative Real-Time PCR, or qPCR. This uses fluorescent dyes or probes, like SYBR green or Tachman probes.
How does that work?
The fluorescent signal increases in direct proportion to the amount of DNA being amplified. The machine measures this fluorescence in real time, cycle by cycle, so you can quantify the starting amount of your target DNA very precisely.
Okay, so we can clone DNA, we can amplify it with PCR. Now, how do we analyze it? Check if we have the right thing, study its expression.
That brings us to techniques like blotting. The original was Southern blotting, named after Edwin Southern.
Southern blotting for DNA.
Correct. You cut the DNA with restriction enzymes, separate the resulting fragments by size, using gel electrophoresis.
Running them through a gel matrix with an electric current, smaller fragments move faster.
Exactly. Then you transfer those separated fragments from the gel onto a solid membrane. And finally, you use a labeled probe, a piece of DNA or RNA that's complementary to your sequence of interest, to find and visualize just that specific fragment on the membrane.
Okay, so it lets you find your specific DNA fragment within a mixture.
Yes. And then, in a bit of scientific humor, when a similar technique was developed for analyzing RNA.
They called it northern blotting.
They did indeed. Northern blotting separates RNA molecules by size and uses a probe to detect specific mRNA transcripts. It tells you about gene expression, if a gene is being transcribed, how much mRNA is being made, and if there are different sizes of transcripts.
Makes sense. Any other blotting?
Well, there's Western blotting for proteins, but sticking to nucleic acids, there's also in situ hybridization.
In situ, meaning in place.
Right. Instead of extracting the nucleic acids, you use labeled probes directly on preserved tissues or chromosomes. Fluorescence in situ hybridization, or FH, uses fluorescent probes.
Ah, I've seen pictures of that, where all the chromosomes are painted different colors.
That's spectral karyotyping, a form of FH. It's fantastic for visualizing chromosome abnormalities like translocations or extrachromosomes.
Very cool. But for the ultimate level of detail, you need the actual sequence, right? The A's, T's, C's, and G's.
Absolutely. Full characterization really requires knowing the nucleotide sequence.
Yeah.
And the foundational method for that was Sanger sequencing.
Developed by Fred Sanger, another Nobel laureate. How did that work? It involved special nucleotides.
Yes, dideoxynucleotides, or DDNTPs. These are modified nucleotides that lack the 3' hydroxyl group.
The hook needed to add the next nucleotide in the chain.
Exactly. So you set up a DNA synthesis reaction, like PCR, but you include a small amount of one type of DDNTP, say DDGATP, along with the regular DNTPs.
Huh.
Whenever the polymerase happens to incorporate that DDATP instead of a regular DATP, the chain stops growing. Synthesis terminates.
So you end up with fragments of different lengths, each ending where that specific dideoxybase was incorporated.
Precisely. You do this for all 4 bases, A, T, C, G, in separate reactions, or nowadays using fluorescently labeled DDNTPs in one reaction. Then you separate the fragments by size, usually using capillary gel electrophoresis. The order of the fragments tells you the sequence.
A very clever method. But things have changed dramatically since then, haven't they, with next-generation sequencing?
Oh, completely. Around 2005, NGS technology burst onto the scene. They took a totally different approach. Instead of sequencing one DNA fragment at a time in a capillary, they use massively parallel formats.
Millions of reactions at once.
Millions, even billions. Techniques like Illumina's sequencing by synthesis, SBS, monitor the addition of fluorescently labeled nucleotides in real time across countless DNA clusters simultaneously.
And the result was?
A massive increase in speed and a dramatic drop in cost. Sequencing that used to take years and cost fortunes became possible in days or weeks for a fraction of the price.
And it didn't stop there. There's third-generation sequencing now.
Right, TGS. This way focuses on sequencing single DNA molecules, often in real time, without needing amplification first. Companies like PacBio with their SMRT sequencing are examples.
What's the advantage there?
One big advantage is read length. TGS methods can often produce very long reads, tens of thousands, even hundreds of thousands of base pairs long. This is incredibly helpful for assembling complex genomes with repetitive regions. We even have portable sequencers now, like the Minion.
Amazing. The cost of sequencing has just plummeted, hasn't it?
Faster than Moore's law for computing power, especially since about 2007. It's been a true technological revolution.
So now we can read the genome quickly and cheaply. The next logical step seems to be editing it.
Indeed. That takes us into the realm of in vivo manipulation altering genes within living organisms, primarily to understand their function. This is gene targeting.
And the classic approach here was making knockout animals.
Yes, typically knockout mice. You engineer the mouse so that a specific gene is intentionally inactivated or deleted, creating a loss-of-function mutation.
And then you see what happens.
Exactly. By observing the phenotype, the physical or metabolic changes in the knockout animal, you can infer the gene's normal function. The example of the obese lep knockout mouse, which lacked the leptin gene, was a classic demonstration of this gene's role in appetite control.
But what if knocking out the gene is lethal early in development? You can't study its function in the adult animal then.
A major problem. That led to the development of conditional knockouts. The Cre-lox system is the most common way to do this.
Cre-lox? Sounds cryptic.
It's quite elegant, actually. You engineer your gene of interest, so it's flanked by specific short DNA sequences called LOXP sites. Then you breed that mouse with another mouse that expresses an enzyme called Cre-recombinase.
Okay.
Cre-recombinase specifically recognizes those LOXP sites and cuts the DNA between them, effectively removing or inactivating the flanked gene.
But how is that conditional?
The trick is controlling where and when Cre-recombinase is expressed. You can put the Cre gene under the control of a promoter that's only active in certain tissues, like the liver, or only active at a certain developmental stage, or even inducible by a drug.
So you can trigger the knockout only in the tissue or at the time you want to study it.
Exactly. It bypasses embryonic lethality and allows much more specific functional analysis.
And the opposite of a knockout would be a knock-in.
Right, transgenic animals. Here, instead of removing a gene, you're adding one, a transgene, or modifying an existing one, maybe to overexpress it or to insert a human gene into a mouse to create a humanized model for studying disease.
Okay, so knockouts and knock-ins were the mainstays. But now there's something even more precise, gene editing.
Yes, gene editing technologies, particularly the CRISPR-Cas system, have revolutionized this field again.
CRISPR, we hear about it all the time. It also came from bacteria.
It did. It's originally part of a bacterial adaptive immune system against viruses.
How does it work for gene editing?
The core is a Cas enzyme. Cas9 is the most famous, which acts like those molecular scissors we talked about earlier. But instead of recognizing a DNA sequence itself, Cas9 is guided to a specific target DNA sequence by a short RNA molecule, the guide RNA.
So you can program the guide RNA to match almost any DNA sequence you want to cut.
Pretty much. There's one small constraint. The target DNA sequence usually needs to be immediately followed by a short, specific sequence called the PAM, or protospacer adjacent motif. Cas9 recognizes the guide RNA and the PAM site to make its cut.
And once it cuts the DNA,
the cell's natural DNA repair mechanisms kick in. They can sometimes make errors leading to inactivation of the gene like a knockout. Or, if you provide a DNA template alongside the CRISPR system, the cell can use that template to repair the break, allowing you to insert a new sequence or correct a mutation precisely.
And this is much faster than the older methods.
Significantly faster and arguably easier. Making a knockout mouse using CRISPR might take around 6 months, compared to maybe 18 months or more, using traditional embryonic stem cell methods. It's efficiency and precision combined.
It really ties everything together, the cutting, the targeting.
It does. It builds on all the knowledge and tools that came before.
Okay, let's try to wrap this up. We've covered a huge amount of ground.
We really have.
We started with the fundamental tools, restriction enzymes to cut DNA, ligase to paste it, and vectors like plasmids to carry it.
Then the strategies for cloning, building genomic and cDNA libraries to catalog genes, and the huge impact of PCR for amplifying specific sequences rapidly.
Followed by the analysis techniques blotting like Southern and Northern to find specific molecules, and the sequencing revolution from Sanger through NGS and TGS that lets us read the genetic code with incredible speed and accuracy.
And finally, the in vivo manipulation, creating knockout and knock-in animals to study gene function, and now the precision of CRISPR-Cas gene editing to make specific changes within living cells or organisms.
These aren't just lab techniques, are they? They underpin so much of modern biology and medicine.
Absolutely. Genomics, forensics, clinical diagnostics, biotechnology, drug development, they all rely heavily on this recombinant DNA toolkit. The progress in just a few decades has been staggering.
It really is stunning. From figuring out how to cut DNA in 1971 to now having tools like CRISPR that allow us to contemplate editing the genomes of organisms, potentially even fixing genetic disorders, the pace is breathtaking.
It raises profound questions, too.
Which brings us to a final thought. Given the power of something like CRISPR-Cas, the source material touches on this deep ethical consideration. Even if we can eliminate devastating genetic diseases by altering the human germline, the DNA passed to future generations, should we? Do we have the wisdom, the right, to make such permanent changes?
That's the multi-billion-dollar question, isn't it? That technology often outpaces our ethical frameworks.
Something definitely worth thinking about. Thank you for joining us on this deep dive into the world of recombinant DNA technology.